In the first part of the class, we talked about B cell development in bone marrow. And we talked about mechanisms which underlie B cell production and especially production of antibody genes in bone marrow. Now we're going to focus on what happens for B cells after they complete their maturation and they get graduated from bone marrow. Here I have a B cell which has IgM and IgD at its surface, and it's been released from bone marrow. Well, most of the B cells which are produced by our bone marrows never get to encounter the antigen for which they are specific. We simply might not get infected or exposed, for instance, to pathogens which contain a particular type of antigen or epitope for which a B cell is specific. So those B cells have a limited limited lifespan from a few weeks maybe to a few months. But if a B cell gets exposed to the antigen for which it has specific antibodies, then that B cell gets activated and starts proliferating. A process which is called clonal expansion, and it's called clonal expansion because after it proliferates, each of these newly generated B cells is exactly a clone of the original B cell. So it has the same DNA molecule and it produces the same antibody with the same specificity. After B cells are generated by clonal expansion, different fates wait for them. One of the things which happen for these B cells, and we will see this in more detail in a lymph node in the next few slides, is that the B cell might get differentiated into a particular type of cell which is called a plasma cell. And plasma cells are funny looking cells. They have a sort of very big Golgi apparatus and an eccentric nucleus, and they are very good at producing proteins. What is the protein that they produce? Well, the IgM molecules. Basically, the plasma cell is the producer of the secreted form of B cell receptors. So the B cell receptors are the membrane-bound form of antibody, and the secreted form is the free-floating antibody molecules. So IgMs are produced by or secreted by plasma cells, and they take a pentameric form, so they have multiple antigen binding sites. IgD is also present at the surface of original B cell and also plasma cell, but usually it doesn't get secreted. Although very, very low levels of secreted IgD is present in plasma, but nobody actually knows what's the function of those secreted IgD molecules. And the levels are nonetheless very, very low. So this is a process which happens for B cells if they do not have any help or any signal from T lymphocytes. What happens for B cells if they get some help or some signal in the form of a cytokine from T cells is a bit more complicated. Some of the T cells which receive some cytokines from T cells undergo what is called a class switching. Class switching is production of new classes or new isotypes of antibodies, different from the IgM and IgD which was present at the surface of B cells in the beginning. So the cells which have undergone class switching can again differentiate into plasma cells, and these plasma cells can start producing antibodies, but this time the antibodies which they produce are different isotypes of antibodies compared to what we had before. They can produce IgGs or IgA or IgE, and which one of these is produced is completely dependent on the type of signal that T cell gives to B cells. And we will talk about this in more detail in the next classes. Another thing which might happen for this B cell is that it might start producing better antibodies. And what do we mean by better antibodies? Well, an antibody which has a higher affinity for antigen. And this can happen if the B cell starts sort of tweaking the gene which produces the variable region of the antibody. By introducing some sort of tiny point mutations in that region, B cell might be able to produce antibodies with a better affinity. So that's another thing which might happen. And another fate that awaits some of these B cells is that they might differentiate into so-called memory B cells. So these are cells that remain in a quiescent state. They distribute all over the immune system, all over the secondary lymphoid tissues, and they wait for the next antigenic encounter. So these things are things which happen if we have some help from T cells. And by the way, these different fates are not mutually exclusive. so they can happen at the same time. So a lot of cells which have undergone class switching at the same time, they undergo affinity maturation. And some of these cells that have undergone class switching and affinity maturation, they do not become plasma cells, but they remain in a quiescent state and become memory B cells. So unlike the picture that I'm showing you here, unlike the flow that I'm showing you here, these three different things can happen at the same time. So this is basically the workflow of B cells after graduation. Now let's look at this processes in a bit more detail by including the organs that B cells go and do these things inside those organs. So let's start from the fetal period. B cell production, or basically generation of blood cells, well, it starts during the fetal period. And liver is the main hematopoietic organ during the fetal period. So there are fetal hematopoietic stem cells, and some of these stem cells become B cells. They undergo the pro-B, pre-B, and immature B stage, and then finally the mature B stage. But the mature B cells which are generated here, they're a little different from B cells which are called conventional B cells that will be produced after basically birth in an adult person. They are called B1 cells. What is the difference? Well, we talked about two different mechanisms for generation of antibody diversity. One was combinatorial diversity and the other one was junctional diversity. The B cells which are produced during the fetal period from the fetal hematopoietic stem cell, they lack a lot of enzymes which are responsible for junctional diversity. So they sort of have a lower diversity in their repertoire. They only enjoy from combinatorial diversity. So B1 cells are kind of limited in terms of different types of antibody molecules that they produce. So that's one difference. The other difference is that when B cells mature after birth from bone marrow, they usually like to go into particular tissues and organs. And two very popular destinations for B cells are lymph nodes and spleen. B1 cells, however, do not like to go there. And they prefer to go to another location, and that's the peritoneal cavity and mucosal surfaces. So there are two major differences. First, they have basically only combinatorial diversity. They don't show a lot of junctional diversity during their development, so their diversity is limited. And the second thing is that the destination for these cells is a little different from conventional B cells that will be generated afterwards. So they usually go to peritoneal cavity and mucosal surfaces. And this is very important. This is a location that a lot of antigenic encounters or a lot of encounters with microbes take place. So they go there and basically try to sort of generate a first line defense there. And this particular B cells or B1 cells which go to mucosal surfaces or peritoneal cavity, they have a particular ability. They have the ability to self-renew and keep their population intact in the absence of stimulation with antigens. So if we look at the B cells which have in our mucosa or peritoneal cavity in an adult person like me, for example, I'm 45 years old, so it's been 45 years since my fetal period and since the time that my liver has been producing B cells. So it seems that considering the limited lifespan of B cells, well, I should not have any B1 cells left in my peritoneal cavity or mucosal surfaces. But if you look at an adult person, if you look at the mucosal surfaces of my gastrointestinal tract or your gastrointestinal tract or peritoneal surfaces, you still see that there are a lot of B1 cells there. Where do these come from? Well, liver is not producing blood cells anymore, so where do these come from? They come from self-renewing capability that these cells have. So they sort of renew their population. They have a sort of stem-like ability so that they remain there for the entire life of a person. And this happens in the absence of antigenic stimulation. Another thing is that these B cells can start differentiation towards plasma cells again in the absence of antigenic differentiation, antigenic stimulation. They generate some plasma cells that produce antibodies, mostly of the IgM class, a limited level of some other classes. And these antibodies are called natural antibodies. Why are they called natural antibodies? Well, by natural here we mean that they are there, even if there are no antigenic stimulation. They are there even if there are no infection with any microbes or anything. So they kind of do it on their own. There are some controversies about the way that these cells are activated. Some scientists believe that there is a level of antigenic stimulation by microbes in the microbial flora of the gastrointestinal tract, but some others dispute that. They say that these cells are able to expand and to differentiate and to plasma cells and produce antibodies in the absence of any stimulation. Nonetheless, even if there is an antigenic stimulation, it's in the absence of any sort of infection or disease. So it's basically leaking or leakage of some antigens from the GI tract, for instance. So they produce antibodies, and these antibodies are always there. One other thing about these antibodies is that these antibodies can bind to a lot of different forms of antigens, mostly microbial polysaccharides and lipids, so they are not very specific. The other thing is that these cells do not produce memory B cells. Well, doesn't really make sense to make memory B cells. There is nothing particular stimulus, no particular stimulus in here to keep a memory of. They're doing it all the time without any particular stimulation. So they produce antibodies which can bind to a lot of different things. They're not very specific for a very particular molecular shape, and they do not produce B cells. So this raises a question. Are they really part of the adaptive immune system? Or they are part of the innate immune system? Because the thing that differentiates innate and adaptive arms of the immune system, there are two things, memory and specificity. We say that innate immune system or innate immune responses do not have it, and adaptive immune responses do have it. Well, for this particular group of B cells, they don't produce memory, so they lack one of those features. And the antibodies that they produce are polyspecific. It's called polyspecific. They can bind to a lot of different uh, uh, microbial sugars, for instance. So they're not very specific. So they kind of are in a gray zone between innate and adaptive immune defense. Uh, well, we separate immune response into innate and adaptive, but there are things which are sort of in between and in the intermediate region or in the gray zone between these two. And these B1 cells are one of those. We will see that there are a subgroup of T cells which act in a similar fashion. We will see it in the next class. So we do have cells which are not completely innate, they are not completely adaptive, they are somewhere in between. And it's believed that they provide the first line of defense against a lot of microbial antigens. So that is the story for B1 cells. Sometimes I compare them. Well, we compare the B cell with a medical student who has graduated from university. Well, sometimes I compare B1 cells with MDs, with doctors, or with other medical professionals, nurses, other medical professionals. That's in that sort of like to go to parts of the world which require medical assistance sort of in an urgent fashion. Well, and those are I don't know, countries with civil wars or poverty-stricken regions of the world, or parts that have been struck by natural disaster. So you've probably heard of Médecins Sans Frontières, the Doctors Without Borders. So those are medical professionals that do not go to big hospitals or very sort of advanced medical centers, or they do not sit in their office. For the patients to come to them, they go to regions that require medical assistance, not very nice regions. There is probably war or poverty or famine, but they provide their services there, and actually it can be extremely efficient. It can save a lot of lives if you go to those regions. So these B1 cells are kind of like Médecins Sans Frontières people. They don't go and sit in big hospitals or medical centers or their offices as the conventional B cells will do. They go to regions that require a first line of defense to mucosal surfaces or to peritoneal cavity or pleural cavity. Those are regions that sort of are more likely to experience microbial invasion or microbial antigens. So they kind of behave like that. Okay, what happens for B cell development after birth? Well, after birth, bone marrow is the major hematopoietic organ, and we have bone marrow hematopoietic stem cells that undergo differentiation towards different lineages, including lymphocytes and including B lymphocytes. These cells enjoy combinatorial diversity and junctional diversity, and they produce the so-called conventional B cells, which have a very vast antibody repertoire. They have a high level of diversity. And these B cells like to go to particular regions of the secondary lymphoid tissues. So to lymph nodes, to spleen, and also to mucosal surfaces. But usually when they go to mucosal surfaces, they go to so-called MALT or MALT mucosa-associated lymphoid tissue. So we're going to see what happens for these B cells. Well, compared to B1 cells, which go to sort of frontiers of antigenic encounter, these B cells go to well-organized medical centers with a highly organized structure like lymph nodes and spleen, and do their work there. So here is a picture of a lymph node. I'm going to show you what happens for a B cell in a lymph node. The story of the spleen will be kind of similar. Well, this is a picture of a lymph node. From previous classes, you probably know that it has a cortical region and a medullary region and an area in between. Well, I'm going to use a highly schematic picture for a lymph node. It separates these three regions. Well, nobody's lymph nodes is going to look like this, but for the sake of simplicity, I've separated them by sort of these elliptical figures. So we have the cortex and the paracortex and medulla. Then we have the so-called afferent lymphatics, which bring the lymph to the lymph node. And then we have the efferent lymphatic, which takes the lymph or drains the lymph away from the lymph node. The lymph node also has an artery and a vein because it requires blood supply and oxygenation. And also this artery is the way that newly generated or newly graduated lymphocytes can enter the lymph node. And there is a particular type of venule in the region between cortex and paracortex, and this venule is called high endothelial venule. So it's a type of endothelium that has a big sort of endothelial cell called high endothelial venule. And this is the gateway for the newly graduated B cells and T cells that allows them to enter the lymph node tissue. So imagine that here I have a B cell which has been just graduated from bone marrow. It enters the lymph node, comes inside, it circulates inside the lymph node, and it reaches the venule. After it goes through the arterioles and capillaries, it reaches the venule. And this venule has a particular endothelium, as I mentioned, and it allows the B cell to leave blood and enter the lymphatic tissue. So when it leaves the blood circulation, it goes towards the cortex because there is a particular type of cell in here which attracts the B cell, and we will see that cell in a few minutes. The same thing happens for T cells, but when T cells enter the lymph node and they appear at the high endothelial venule, they leave blood circulation and they go towards the paracortex. So the cortex will be the B cell region and the paracortex will be the T cell region. So, well, it's not going to be a single B cell. A lot of B cells will leave blood circulation and they sort of gather around each other. Well, in reality, they gather around some cells which have been attracting them. And they give rise to what is called a B cell follicle. And this is called a primary B cell follicle. So it's sort of a gathering or a collection of newly generated B cells that have left the high endothelial venule and there is a sort of an aggregate of B cells in here. And it's not just in there, it could be in here, in here, in here, in multiple locations. In the cortex, you will have these primary B cell follicles. So what we're going to do now is to focus on one of these follicles and see what happens for it if it sees the antigen. Okay, so I zoomed on in this region of the cortex, and here I have a primary B cell follicle. Before we see the antigenic encounter, I should show you this particular type of cell, which is the reason that B cells have aggregated in that region. And it's called a follicular dendritic cell. It's a cell which has a lot of branches, so it's called dendritic, and it's located in the follicles, in the B cell follicles. And it's not a single cell, there are multiple of them. I'm showing only one of them. And these dendritic cells are very different from the classical or conventional dendritic cells, which are part of the antigen presenting system and present antigen T cells. So only they have a similar morphology, but in terms of function, they're completely different. So we should not confuse them with the conventional dendritic cells which activate T cells. So these follicular dendritic cells produce a sort of a chemokine which attracts the B cells, and then the B cells will go there and form the follicle. Now what happens is that an antigen enters. Where do antigens come from? Usually from afferent lymphatics. So they enter from the afferent lymphatic and then go to the cortex. And the follicular dendritic cell has an ability to catch these antigens. So it has some sort of receptors for them. By the way, these antigens, usually they come from the periphery. So if, for instance, I have a lesion in my hand, that antigen might enter the lymphatics and enter the lymph nodes in the antecubital region or in the axillary region. So sometimes antigens come by themselves, and sometimes antigens get captured by antigen-presenting cells which are in the tissues. You've probably heard about Langerhans cells, which are in the skin, and they're kind of an APC, kind of an antigen-presenting cell. They capture the antigen and they carry it to the lymph node and bring it to the follicular dendritic cell. So that's another possibility. Anyway, either the antigen itself or carried by a cell enters the cortex. It's captured by the follicular dendritic cell, and this follicular dendritic cell starts showing this antigen to B cells. What happens next depends on whether we do have a B cell with antibodies specific for this antigen or we do not. If we happen not to have any B cells with antibodies specific for this antigen after some time, it's washed away by the lymph flow, and it goes out and leaves the lymph node through the efferent lymphatic. But if I happen to have a particular B cell or some B cells with antibodies specific for this antigen or specific for its antigenic determinants, then these B cells start migrating towards the center of the follicle, and then there they start proliferating. And I've kind of shown this in a sort of a bigger size because these cells that are proliferating, they kind of are bigger than the original B cells. They are called immunoblasts. So start proliferating. Some other things also happen in here. When some B cells start proliferating in the center of primary follicle, this follicle is no longer called primary, it's called a secondary follicle. And the secondary follicle is the type of follicle which has this so-called region for proliferation of B cells. It's called the germinal center. And this is something which, when you look at a lymph node under a microscope, you can use it to distinguish between primary and secondary follicles. So primary follicle is just an aggregate of B cells without any germinal center. It has either not received any antigenic stimulation, or if it has received some antigens, none of those B cells have been able to respond to that antigen. But a secondary follicle is a follicle with germinal center. It has basically a region in which B cells are proliferating. So if I look at this particular picture from a lymph node, you can probably see some primary follicles, like this one in here. Or let me see, maybe this one in here. It's a primary follicle. I don't see any germinal center in here. This one is a primary follicle. I'm not seeing any germinal center in here. Maybe this one is a primary follicle. But this one is a secondary follicle. It has a lighter staining region in the center, which is the germinal center. And it means that this is reacting to some particular type of antigen. This is a secondary follicle. Again, this is a secondary follicle. And it's important for a pathologist to be able to differentiate between these two, because if there are no secondary follicles, it means that the lymph node is not reacting to anything. But if there is a secondary follicle, it means that it's reacting to something, and this could be medically important. All right. So here I'm focusing on a secondary follicle, sort of zooming on, sort of a schematic zooming, not real zooming under a microscope. And as I mentioned, this region in the center is called the germinal center. B cells which are on the periphery are the quiescent, non-proliferating B cells. And this region is called the mantle zone. And this region in the center is the germinal center, as I mentioned. And this is where B cells are proliferating. So the B cells which have been proliferating, or which have responded to the antigen, can have different fates. And I showed you some of these fates in one of the previous slides. These fates are completely dependent on the nature of the antigen to which B cell is responding. If the antigen that I'm having in here is a polysaccharide or a lipid or a sort of glycolipid, anything other than protein, then the fate of these B cells is relatively simple. These B cells, most of them differentiate into plasma cells, which are antibody-producing cells. And if I put this back in the cortex, these plasma cells migrate to the medullary region of the lymph node, and they start producing their antibodies there. What is the antibody that they produce? Well, an IgM class antibody. What happens for that antibody? Well, you know that there is a flow of lymph from afferent to efferent. So that flow of lymph takes the IgM molecules with it, and it enters the lymph. And we know that lymph finally enters blood, so the IgMs will end up in the blood circulation. So this is what happens for B cells if the antigen which has stimulated them is a non-protein antigen. Differentiation into plasma cells and production of IgM. This type of response is called a T-independent response. And as the name implies, it's a type of response that does not use any help or any signal from T lymphocytes which are present in the paracortex. Why? Because T cells only like to respond to protein antigens. Their receptors in general cannot respond to sugars or lipids or those kinds of things. So if the antigen that I have is purely a polysaccharide or a lipid or a combination of these two, but not protein, but lacks a protein part, the T cells do not get involved. Another fate is when the antigens do have a protein component, and this is the case for most of antigens. If the antigen has a protein part, in addition to stimulating B cells, it can also stimulate T cells. Well, we know that it's not a single molecule of antigens, so maybe hundreds of them enter the lymph node. So some of them get stuck in the cortical region, but some of them can come to the paracortical region. And here we do have some cells like dendritic cells, classical dendritic cells, not like the follicular dendritic cell, or maybe macrophages, which can capture these antigens and break these antigens into some peptides and load those peptides at the surface of an MHC molecule. This is what antigen-presenting cells do. They capture an antigen, phagocytosis or pinocytosis. They degrade that antigen in their lysosomes. They break it down, and then they put the resulting peptides at the surface of MHC molecules. They present it to T cells, and then the T cells start proliferating and then differentiating. And then when the T cell differentiates, and we will see this in more detail in the infectious immunology class, they start producing cytokines. And these cytokines can influence the B cells in the cortical region. So there is a sort of a proximity in here. These cells are in the paracortex, these guys are in the cortex, but they are very close to each other. When B cells get influenced by the cytokines produced by T cells, we will have a more complete or more sort of advanced type of antibody response, and it's called the T-dependent immune response. What is a T-dependent response? Well, B cells can have more complicated fates in here. Again, some of them might be differentiating towards plasma cells and producing IgM. So that's still possible. But another possibility is that some of these B cells undergo class switching and produce plasma cells which are able to generate new classes of antibodies like IgG and A and E. And as I mentioned, which class is produced depends on the type of cytokine which is generated by T cell in here. Also, these B cells, some of them can start introducing tiny mutations in the gene which produces the antibodies, the variable region of the antibodies, producing higher affinity antibodies, and some of them can become memory B cells. And I showed you these different phases in the previous slide, but here I'm putting them sort of in the histological context or in the context of the tissue. So these are more complicated things, and these are called T-dependent humoral immune responses. All right, so here I want to end this part by making a connection between two different concepts that we hear a lot: T-dependent and T-independent responses, and primary and secondary responses. So these are two highly related concepts. What is the T-dependent and T-independent responses? Well, we just saw that if the nature of the antigen is a non-protein antigen, T cells do not get involved, and the response that we have is mostly an IgA production of IgM producing plasma cells. So it's called T-independent, but if we have involvement from T cells, then we have other fates like generation of memory B cells and class switching and affinity maturation, that kind of thing. So that's the T-dependent and T-independent. Another concept that we have in immune response in general and in antibody responses in particular is the primary response versus secondary response. So when we say primary and secondary, what comes to mind is that, okay, primary response is the response that we get the first time. When we see the antigen for the first time, well, the response is primary. When we see the antigen for the second time or maybe the third time, it's called secondary response. Well, it's not completely like that. Primary response and secondary response are not merely reflecting the first time versus the next time of antigenic encounter. Primary here mostly means sort of primitive, not very complicated, not very sophisticated type of response. And secondary means more complicated, more sophisticated type of response. And the evolution from primary response to secondary response is a process which requires involvement from T cells. In other words, if I have a type of antigen which does not involve T cells, like polysaccharide antigen or lipid antigen, regardless of how many times we get exposed to that antigen, our response will always be primary. So it's mostly IgM, not very much of other classes, not much memory B cell formation, not much affinity maturation. So this is called the primary response. And even the second time, again, my second encounter with the antigen, my response will be primary. The third time, again, my response will be primary. So that's the meaning of primary in here. But if T cells get involved, well, in the beginning, in the first antigenic encounter, because T cells need some time to get involved, well, B cells produce mostly IgM and it takes time, but then after involvement of T cells, the response gets sort of more sophisticated, more advanced, and it entails generation of new class of antibodies and memory B cells and affinity maturation and so on and so forth. So this is the relation between these two concepts. All right, so this was the part for B cell activation, and then the next part will be antibody function.